Today, I want to tell you about a cool technique called hydrogen-deuterium exchange mass spectrometry, or HDX-MS. It can be used to tell you information about solvent accessibility, so which regions of, say, a protein are exposed to water or whatever other solvent you're in, as well as secondary structures and binding sites of a protein. And the really cool thing about HDX-MS is that it's in solution, so you don't have to get the protein to freeze, you don't need to get it to crystallize, you get it monitored in its natural environment and look at its dynamics. It's not as commonly used as other techniques, in part because it uses some specialized equipment, but I used it in grad school and got some cool results from it, and so I'll show you that as an example and walk you through how we can interpret this data and what's involved in an HDX-MS experiment, what it can tell you, that sort of thing. So let's dive in. We'll get a lot more into the details, but you can think about it kind of like doing a cannonball into a pool and looking to see where you get wet. The parts of you that were protected by a swimsuit are going to be dry, as well as the parts of you that were kind of hidden in the interior of your cannonball, assuming that you don't like open up and all that stuff. But if you think about just kind of conceptually, the parts that are protected from getting wet will be drier than the parts that got wet. The parts that got wet, well, now they have the water weight and they're going to be heavier. Similarly, we can stick a protein in a bath of what we call heavy water, deuterated water. We'll get into this in a minute, but deuterium is a heavier isotope, so a heavier version of hydrogen. And that deuterium can go and swap out for hydrogens in the protein if those regions of the protein are exposed to the deuterium and they are not tied up in forming other interactions. Over time, more and more of those hydrogens in the protein will get swapped for deuteriums. And then if you cut up those pieces and weigh the pieces of the protein, the pieces that had exchanged the hydrogens for deuteriums will be heavier. And you can use a technique called mass spectrometry in order to detect the weight of the pieces, compare those to a database, and see which parts of the protein were heavier than others, which could indicate that those parts of the protein were solvent exposed, those parts of the protein that lacked a secondary structure, more in a minute, and things like this, whereas regions of the protein that remained light will be protected from exchange for some reason, such as maybe they're involved in secondary structure, they're binding to a binding partner, maybe they're tied up in the center of a protein, all these various things. And because HDX-MS is working in solution, you can see changes in these dynamics over time and in the presence of different conditions. So we'll get much more into the details, how you interpret the data, all this stuff. But let's start by just going back and thinking about how this exchange is happening, which means we need to go back and think about the structure of a protein. So remember that a protein is made up of a string of amino acids connected together through peptide bonds. These peptide bonds are special. They are resonance stabilized. Basically, there's going to be electron sharing between this oxygen, this carbon, this nitrogen. So these all have to be in a plane. This is going to limit the rotation of the peptide backbone. There are certain angles that are more favored, and these angles correspond to some common secondary structures you see, things like alpha helices and beta strands, which combine into beta pleated sheets. And these are held together through hydrogen bonding, involving interactions between the oxygens and the hydrogens of the nitrogens in that peptide backbone. Note that a peptide bond is a kind of amide bond. So an amide bond is just this functional group. And when you have it in the context of a protein or a peptide, we call it a peptide bond. So this is an amide bond. This nitrogen has a hydrogen that can act as a hydrogen bond donor. Because the nitrogen is more electronegative than the hydrogen, it pulls away electrons, leaving the hydrogen partly positive and the nitrogen partly negative. On the oxygen is more electronegative than the carbon, so it's pulling away electrons, making the oxygen partly negative, the carbon partly positive. You also have a lone pair on the oxygen, so the oxygen on from one amide bonds, one peptide bonds, can form hydrogen bonds with the hydrogen on a nitrogen in another part of the protein or another chain of a protein, and this can lead to hydrogen bond formation. Keep this in mind because that's going to kind of prevent this hydrogen from being swapped out. The protein then folds up further to give you tertiary structure, and then multiple chains can come together to give you a quaternary structure. And so not all proteins will have a quaternary structure, but some will. In both the tertiary structure, you're hiding some regions of the protein because they're going to be kind of hidden towards the center. And then the quaternary structure, you could be hiding parts of the protein because they're going to be involved with interchain interactions. You can also have protection from things like if you're binding to a binding partner, that's going to hide part of you, as well as if you are in a sort of different shape. Because those hydrogen bonds can be broken and reformed in different ways in when you have conformational changes, or basically changes in the structure of a protein that don't involve you actually breaking any real covalent bonds, just kind of rearranging the protein structure. So let's think about how this exchange is actually happening. So remember that a peptide bond, that's a form of amide bond. It's just the name we give to an amide bond in the context of a peptide or protein. The hydrogens in this amide bond are going to be the chief site at which we're able to detect the exchange of hydrogen for deuterium. You see hydrogen other places in a protein, such as attached to carbons, say like your alpha carbon or other carbons in the side chain, as well as in some of the side chains that have amine groups or that have carboxylic acid groups. These can exchange hydrogens as well. But as we'll see, these hydrogens on the oxygens and the nitrogens are going to exchange too quickly in your side chains, and those that are attached to carbon are going to exchange too slowly. The one at the amides happen just right. These are going to be slower than the ones in, say, a primary amine or something like that in your amino acid side chain. Because the peptide bond, this amide bond, is stabilized by resonance, so these are going to exchange more slowly than the side chain nitrogens. These are going to exchange at just the right level so that we're going to be able to detect it, and I'll tell you in a minute about how we'll detect it. Whereas the hydrogens on the carbon, these are too tightly held to the carbon. They're going to exchange very, very slowly, and so that's not going to be useful on an experimental time scale. What's going to happen? So we have protium, which is your normal version of hydrogen. So if you just have a protein in a normal buffer, you have a protein. So a buffer, that's just a word we use for pH stabilized salt water. This is just this word, basically a generic word for a solution. And typically our solutions are going to be aqueous. They're going to be having normal water. They're going to have protium as the hydrogen source. And so protium and deuterium. So protium's your normal hydrogen. Deuterium is your heavy hydrogen. They differ in their number of neutrons. So different elements that have the same number of protons are the same element, but they can have different numbers of electrons and different numbers of neutrons. If they have different numbers of electrons, then we can have ions because the charge of the electrons won't balance the charge of the protons, and so the molecule will be charged. In the normal state, both protium and deuterium are neutral because they have one proton and one electron. However, deuterium also has a neutron, whereas protium does not. So protium and deuterium are going to be isotopes. Sometimes when you have isotopes, when you have different numbers of neutrons, the number of neutrons can be kind of more than the atom can handle. And so it'll spit some things out in the form of radioactivity. Deuterium is not radioactive, so this is not a radioactive experiment, totally safe in that regard. But the deuterium is heavier, and it's heavy enough that you can detect it with a specialized equipment like a mass spec. So we want to keep an eye on these amide hydrogens and see how they can exchange for deuterium. Note that this deuterium exchange is going to be pH and temperature dependent, and we're going to take advantage of this later when we quench the reaction. So we can stop the reaction when we want to stop it and see what's heavier. Imagine you have a race and you want to see what places different people in the race finish, but you don't have a camera you can constantly monitor the race. And so you want to choose a few different times in which to take a picture of the finish line and see who's crossed it. If you were to wait and take it like an hour after the race, that wouldn't be very helpful because everyone would have gone through and you won't be able to tell what order they came in. If you went too soon, well, now you wouldn't see anything. So you want to kind of space things out so that you can get the best sense of the order in which people arrived. Similarly, in an HDX-MS experiment, we're going to take different time points typically, and at those different time points, we're going to stop or quench the reaction. And as you'll see here, basically on your y-axis, this is the efficiency of the exchange. It's going to be most efficient under more basic conditions as well as higher temperatures. So if we get to this low acidity, about 2.7, and a low temperature, we can effectively stop the exchange from happening. So keep this in mind because we're going to get into it later. But the reason why I wanted to show you this now was to show you that we're going to typically be in this pH region of physiological solutions. We're somewhere around 7, 7.4, 8. In these pHs, we're going to be in this more basic region where a base catalyzed exchange mechanism is going to predominate. What you can think of is in water, in deuterated water, in protium water, so your normal water, your heavy water, in both of these cases, you're going to have some amount of the water be ionized. In more basic solutions, you're going to have more of the hydroxide ions, more of the OH minus, and so those are going to be around in order to help catalyze reactions. What can happen is you can have a hydroxide ion that actually goes and acts as a base and steals one of those protons from the amide backbone. This is going to take away the normal hydrogen. Now, that nitrogen is left with a negative charge. It's left without a proton. It has a lone pair. That lone pair can then act as a nucleophile. It can act as a base, steal a deuterium from a deuterated water particle. And this is then going to make the backbone have a deuterium instead of having a protium. And voila, your peptide is now heavier. This exchange is going to be easier if that region of the protein is easier to access for the deuterated water. And if that region of the protein, if that hydrogen is not tied up in the secondary structure formation. This is the typical reaction that's going to predominate at the pHs that we use. But note too that at a very low pH, there's going to be acid catalyzed mechanisms. We're going to be dealing mostly with the space catalyzed mechanism, and it's not crucial to understand the mechanism to understand the interpretation, just so that you know, because chemistry is cool. This is the reaction that we're typically going to see in this HDX-MS experiment. And this also helps you see that when you do the quenching, it's not just that you want to get acidic, you want to get to a very particular pH, about 2.7. You might have to play around a little bit in order to make sure that you get good quenching. And I'll tell you a little bit about some of the controls that you can do in order to correct for any sorts of after exchange effects. Hopefully this will make more sense in a minute. So let's go through the basic idea of an HDX-MS experiment. You have your protein in water. Now you're going to label it. You switch it into deuterated water, so that heavy water. You dump it in the bath. Now what's going to happen is that regions of the protein that are flexible, that are solvent exposed, that aren't involved in secondary structure, those regions are going to exchange more rapidly those hydrogens for deuterium. You're then going to lower the pH and temperature at different time points, take your pictures of the finish line, by quenching the reaction. So remember, we can lower the pH, we can lower the temperature, both of those would stop the exchange. By taking samples at different time points, well, now we have different glimpses into the dynamics of the protein and the relative accessibility of regions. We do this for a number of time points. Then what we want to do is we want to be able to see which parts of the protein were heavier. The way that we're going to typically do this is by cutting the protein up. So you can use a protease, a protein cutter, an enzyme that cuts proteins. So different protein cutters have different specificities. The key thing here is that you need to use a protease that is stable at low pH, that is active at low pH. This limits your options. A typical one that's used is pepsin. This is going to cut up the protein into different peptide fragments. And those fragments will then be lighter or heavier depending on whether deuterium was exchanged in those regions of the protein. So you can use liquid chromatography in order to kind of clean up and separate those different pieces, and then measure the weights of those pieces. Typically, instead of just measuring the weight, what you're doing is measuring the mass to charge ratio using a technique called a mass spectrometer or a mass spec. If you don't cut up the protein, well, now you can just get information about the global sorts of exchange. So see if a protein overall is kind of more or less flexible, more or less open, more or less dynamic, these sorts of things at different times under different conditions. But you don't get the very granular things that you could get if you were to, say, do a local comparison. To do local Hdxms, then you want to compare exchange to the peptide level, which means that you need to do that cleaving. So you do that pepsin cleavage, you do other protease cleavage. There are also more kind of non-specific methods I'll tell you about a little in a second. various methods in order to fragment up the protein, and that'll allow you to do the local exchange. You get the mass to charge ratio of these different pieces, and then what you can do is you can compare them to database to figure out where in the protein those pieces correspond to. And then if there's a structure of the protein known, or now even if there's just an AlphaFold predicted structure of the protein known, you're able to map those changes to the structure of the protein to see which parts of the protein were more or less protected. And again, with both the global and the local, you could be doing this in the presence of different conditions, which would allow for differential Hdxms. So you're comparing differences with results that you get under different conditions, such as with or without a ligand. So a ligand, that's just a binding partner. It can tell you like which regions the protein might be binding to the ligand because those parts are going to be more protected, or if there are conformational changes, so shape changes that occur under the binding of the ligand. So what do these results actually look like? I'll show you some actual results in a minute from my experiments. But you basically get spectra for the individual peptides that show you the extent of the exchange. So you can see that you kind of get this mass to charge ratio, this MZ, mass over charge. And then on your y-axis is your relative abundance. You get a sort of envelope due to things like isotopes of carbon. So you get these sorts of mass envelopes. Then later you can get differences in the envelopes because of differences in the deuterium exchange. Basically, the heavier things are, the more to the right they're going to shift to the higher mass to charge ratio. And so what you can do is you can then measure the change, the movement of the envelope, the sorts of average size, average weight, if you will, of those pieces. And you'll see that as they get deuterated, the curves are going to shift to the right. And so you can do this over different times and for different peptides. Here's an example of what these things might look like. So if you had a fast exchanger, you see readily, this is at like 10 seconds, you already have it all kind of switched over to the heavier form. Whereas if you have a slower exchange, well, now it takes 2 hours in order to get to that same level of exchange. Often it's going to be the relative abundance or the relative intensity on your y-axis. So you normalize these to say the number of hydrogens there are to exchange, so you can fairly compare between the different pieces. You can then plot the same peptide across time points to get peptide uptake plots, showing you the uptake of deuterium under different conditions over time. So you can see that in state A, this peptide was exchanging more rapidly than in state B. And so this can tell you about differences that are happening under these different conditions. What you could do then, if you want to find even more information, you want to get a little more context, you can map this onto known structures, and maybe you might see, oh, that's a region that's expected to interact with blah, blah, blah. And so when we add blah, 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 then we see that it's more protected. That makes sense. Or, I wonder if that's where blah, blah, blah binds, because when I add blah, 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 it gets protected. Another FYI that you probably won't have to worry about, but if you want to actually dive into some HDX, you might see a couple of different types of kinetics. The two main ones are EX1 kinetics and EX2 kinetics. EX1, this is going to also be known as correlated exchange, and EX2, this is also known as uncorrelated exchange. EX2 is the sort of more common one. What EX2 means is basically that your refolding is happening much faster than the exchange. So every time the protein kind of breathes, so it's kind of weird to think about a protein breathing. You're like, a protein doesn't have lungs, but a protein is dynamic. So even though we think about it as sort of like a static thing a lot of the times, one of the great things about Hdxms is that it's allowing you to see the dynamic nature of the proteins. And one thing that the proteins do is they kind of breathe. Now, when they breathe, this doesn't mean that they're like totally unfolding or anything. It's just little regional kind of breaks in the protein, not breaking actual covalent bonds, but just kind of those hydrogen bonds. So it's kind of, if you think about you're holding someone's hand for a really long time, you might kind of let go real quickly to get a regrip. The same sort of thing happens with your proteins. Hydrogen bonds can be broken and reform, even in regions of a protein that are kind of more solid in the structure. Especially because they're more solid in the structure, the things around them are going to stay bonded when they breathe open, and so then you're going to have it so that you will quickly rebind as well. But if during that breathing motion, when you broke the hydrogen bonds, if a deuterium stuck in there, well, now you can get that exchanged. So you can think about a protein kind of opening and closing and opening and closing and opening and closing. And remember, these can be dramatic opening closings, or they can be just little localized little breaks in the protein structure. And again, breaks not in terms of breaking the backbone, but just breaking some of those interactions, making it so that the deuterium can exchange. When that happens, the protein is going to be kind of like permanently marked. If you imagine that you were doing your cannonball into a pool of paint rather than into a pool of water, what's going to happen is that parts of you are going to get painty. And even if you were just to dip in really quickly, you would get painty. And if you were to stop dipping, you get out of the water, you're still going to be painty. And so the protein, every time it opens up, has a chance to get one of those marks. It has a chance to get a drop of paint on it. And when it gets a drop of paint, well, now it's permanently painted. And so it's going to be higher up. And so in terms of its mass to weight ratio. And so there's no going back. Each time you open up, you kind of get bigger. You get heavier, you get heavier, you get heavier because you keep getting little parts, little regions that are going to exchange. And so one copy of the peptide might exchange at one time point, and then another copy at a different time point, and then the first copy could exchange at a different site within the same peptide. So what's going to happen is you're going to get a single increasingly heavy mass to charge envelope over time. This is your EX2 kinetics. This is the more common one. The other option, or the other main option, is that your exchange is actually happening much faster than refolding. So when we think about EX2, we're thinking about, okay, every time it opens up, you're going to get sort of one exchange or just a couple exchanges. Whereas with EX1, here we're saying, okay, well, it opens up and then it stays open for a while, so everything's going to exchange. Now you kind of get a bimodal distribution of unfolded and exchanged, and not yet unfolded, and therefore not exchanged parts. And so you kind of have these two main envelopes rather than one envelope that's kind of moving together. You have the unfolded that's going to be deuterated as soon as it unfolds, and it's going to kind of stay unfolded, or it can fold back up, but we wouldn't know. And then you have the one that has not yet unfolded, and that's going to be unexchanged. But as soon as it unfolds at least once, you're going to get this increase in the mass to weight ratio, even if it goes back. And so you're going to see over time, you're going to eventually have all of the exchanged, but you're going to have this bimodal distribution rather than having this one moving envelope. Let's talk about some potential problems and controls. So we say we want to do that quenching. We want to take the pictures on the finish line at those set time points. Imagine that your camera had a really, really, really slow shutter speed. So you could have people that are crossing the finish line, or you could have people that kind of go back and go through the finish line. They sort of walk backwards into the race. Maybe waited so long that they kind of just got bored. They're going to go find their family and friends in the stands. So we can think about with a protein, you can have in exchange, where what's happening is kind of more people are crossing the finish line after you take the picture. More deuterium is getting added even after you wanted to quench it. And back exchange, where when you quench it, you're quenching it in normal water. And so what can happen is that some of the deuteriums that had exchanged for deuterium had actually then go and swap back for hydrogens. And this is actually why you can't detect like those hydrogens on various side chain groups, on your oxygens, on your carboxylic acids, on your primary amines, because they're just going to exchange back to water too quickly for you to detect. But we really don't want this back exchange happening in the places where we would expect the deuterium to actually stick around in those backbones. And so for both of these regions, it's important to optimize your quench conditions. Even if you optimize them, you need to make sure to include controls. There are a couple of different types of controls you can do to account for this. There's like an in-exchange control and a back exchange control. For your back exchange control, sometimes you're like 100% maximal, your Dmax. What you can do is you can chop up the protein. So you use that pepsin to digest it, say, and then you label it. So you're labeling it after you digest. This is going to tell you the maximum amount of uptake that can happen at these different peptides. There's also an in-exchange control where you do the labeling after the quench step to see how well the quench step is actually preventing more deuterium from being incorporated, which is going to tell you about how good the quench step is at preventing more water from being incorporated, more normal hydrogens. There are various variations and improvements. So some of these involve doing different ways to digest the protein. So instead of using proteases, you can actually use energy beams to dissociate the protein into different fragments. This can help for things like trying to get more variety in your fragments because these methods don't have as much specificity kind of as those proteases do. There are also various other things that you can do to improve the resolution, so the kind of telling apart the different fragments, different things like that. So really great review article I will direct you to. Hopefully HDX-MS will become more widespread. It's used in a variety of different experiments, analyzing a variety of different proteins, things like receptor binding, things like looking for unwinding of symporters and small molecule binding and expansion dynamics of the dengue virus capsid. So just some really cool stuff. And so another great review article you can look at. Speaking of looking at things, let's look at an example of what I know very, very well. The PhD work I did in Lemore Joshua Torres' lab at Cold Spring Harbor Laboratory. And special shout out to Alad El-Kayam, who was the researcher in the lab who really served as a mentor for me. So thanks, Alad. So what was I studying? I was studying RNA interference, also known as RNAi. The basic idea is that if you think of your essential dogma, you have a gene, so that's DNA, that gets transcribed into messenger RNA, and that gets translated into a protein. You also have some genes that instead of coding for messenger RNAs that code for proteins, they have RNA products that actually just function as RNAs. And one type of these RNAs are small RNAs, such as microRNAs or siRNAs. And so microRNAs are the main ones that we use as humans and mammals. And so what these microRNAs do is they bind to a protein called argonaute, and then the protein argonaute uses those microRNAs as guides to go and target various messenger RNAs that have the instructions for making other proteins. Using that microRNA as a guide, argonaute directs the sorts of repression of those messenger RNAs so that less of the proteins get made. In a little more detail, so you can understand why we want to use HDX-MS for this experiment, argonaute binds to a guide. So again, this is like your microRNA. This is going to give you the core RNA-induced silencing complex, or RISC. In that microRNA, so the microRNAs are typically about 22 nucleotides long, and in the beginning region, there's this sort of seed. And this seed is going to be about 16 nucleotides, and this is going to serve as the sort of guiding sequence. This sequence is going to have complementarity to a sequence in the messenger RNA of the things it's going to target. So you can think of it kind of like the microRNA acting to put an address into RISC, like a self-driving car, like telling it where to go, and it's telling it to go to messenger RNAs that have a complementary sequence to that. Typically in the 3' UTR, so the part of the mRNA that's after the part that actually codes for the protein, there's some sort of regulatory information, and argonaute can bind there and have effects. What kind of effects? Well, when argonaute binds, if it has more complementarity than just the seed, if it has like full complementarity, this is often the case with siRNAs, then argonaute can actually cut that messenger RNA and lead to its degradation. Or argonaute, in the case of partial complementarity, which is more common in us, it can bring in deadenylation complexes, so to kind of remove the poly A tail, and decapping complexes to remove the cap. All these can lead to translational repression or mRNA degradation that can decrease the amount of protein that gets made. Argonaute then can release that messenger RNA once it's done, and then go and seek out additional targets. So it can catalyze the destruction of a lot of different messenger RNAs. Argonaute's really, really cool. But in order to do this over and over again, argonaute needs to be able to release the target messenger RNA. And there were a lot of unknowns about how this process occurs. This is what I looked into in my grad school work. More specifically, I was looking at the eukaryotic insertion, which is a region of the protein that is found in eukaryotic argonaute proteins, but not prokaryotic argonaute proteins, and it contains a number of potential phosphorylation sites. Previous work had found that these sites, or at least some of these sites, were phosphorylated, and they were phosphorylated by a kinase called casein kinase 1 alpha, CK1 alpha, and this phosphorylation was affecting the binding to targets. So in my PhD work, I tried to figure out more how this was actually happening at the mechanistic level. What was happening in the protein to account for these effects? And one of the techniques I used was Hdx-MS. And so I'm going to tell you about how we use HDMX to kind of decipher part of what was going on. If you want to know more about the details, you can check out our eLife article that was published in 2022, open access so anybody can read, and I will provide a link. If you want to study a biochemical system in mechanistic detail, so actually really dissect what's going on, you need to be able to control the conditions. You need to be working in vitro. And so I needed to develop an in vitro system in order to detect the phosphorylation and test different conditions and things like this. And so I took argonaute and I purified the argonaute protein, and I took the kinase that was responsible, the CK1 alpha, and I purified that, and I mixed them together, and I didn't see any phosphorylation. And I'm like, uh-oh, this is not a good start to your PhD project. Then I thought, okay, well, maybe if I add some guide, it'll get phosphorylated. I added guide, no phosphorylation. What if I add target? Well, for target, this is just kind of a piece of the RNA that is going to serve as a fill-in for the messenger RNA, so that portion of the messenger RNA that actually binds to the microRNA. So you can see you have the seed region, as well as additional pairing at the supplementary region. When I added this target, well, now I see the phosphorylation of argonaute. Phew, my PhD project is still alive. This also told us some important things because it was saying that we needed the binding to the target in order to promote the phosphorylation. One potential hypothesis as to why this might be was that binding to the target causes conformational changes. So conformational changes referring to changes in the shape of the protein, potentially in the eukaryotic insertion. So if you have binding to the target changes the eukaryotic insertion, perhaps making it more accessible or and somehow in a better conformation for phosphorylation by CK1 alpha. So this is a hypothesis, but how could we actually test it? To understand this, I need to first tell you a little bit about the structure of argonaute. And so this is the structure of argonaute that was solved by X-ray crystallography. So it's a technique where you can get atomic level detail of a protein structure. This is a level of protein detail, structural detail that we cannot get from Hdx-MS. Instead, with Hdx-MS, we're getting information that we put on top of this sort of structural data. But the benefit of Hdx-MS is that it's happening in solution. You don't need to actually freeze the protein. You can get information about regions of a protein, say that are not visible in X-ray crystallography for reasons I'll talk about, or regions that kind of are dynamic, and so they don't have a fixed structure, or they're actually changing over time. But to help orient us to what we do know about the protein already going in, you have these four main domains, the N, PAS, mid, and PW domains, as well as two linker domains, L1 and L2. Argonaute is then going to hold the small RNA. So again, that's like your microRNA, your siRNA. It's going to like kind of wind through the protein. So it's going to have one end, the 5' end of that microRNA held in the mid domain, and the 3' end held in the PAS domain. So if you want to think about it a bit like a duck, as Lemur likes to do, you have the beak of the duck in the PAS domain holding onto the 3' end of the RNA. What about the eukaryotic insertion? Well, that's actually in this part of the protein here. You can see on either side of the eukaryotic insertion these ends here, but the eukaryotic insertion itself is actually in this region that we can't visualize with the crystallography, making Hdx-MS a really great option to see what's going on. But why can't we see what's going on in crystallography? X-ray crystallography is great for a lot of things, giving you really high resolution data for regions of a protein that are stable, for regions of a protein that are really sturdy. Because with X-ray crystallography, you have to actually get the protein to kind of freeze and freeze on its own volition. So basically, you have to get all these copies of the protein to stop moving around and freeze in the same orientation. Then you shoot X-rays at them. The X-rays are going to interact with the electrons in those protein molecules or whatever other molecules. You can do this nucleic acids and things. But the X-rays are going to interact with the electrons in those molecules. Those electrons are going to basically scatter those X-rays, and those scattered X-rays are going to interact with one another. They can build up or cancel each other out. This is going to give you a diffraction pattern, this series of spots. And then crystallographers can work backwards from that diffraction pattern, from that series of spots, in order to figure out where the electrons were, this electron density map, and then this sort of meshy thing. Then what they can do is they can actually build the atomic model into that map. And so they actually go and they place those sticks and those loops and all this stuff into this meshy thing, this map. And the map is the actual real data. And this is just the model that's coming from the map. In order to actually place the model into the map, you need to be able to see the density, see the map. And there are regions of the map that, because that region of the protein is too flexible, it's moving around too much, it has too high of a what we call B factor or temperature factor, that's not going to have a visible density. And so you're not going to be able to actually map into the region. And so in these really flexible regions, those atoms are often left unmodeled. And this is the case of the eukaryotic insertion. This region is probably too flexible, too dynamic, too something that it's not showing up clearly in our map, and therefore it can't be modeled in. So when Elad went to solve the structure of the protein, he couldn't see the eukaryotic insertion. When I solve structures of the protein, I couldn't see the eukaryotic insertion, no matter how many kind of manipulations I tried to make to sturdy up the regions on either side and this sort of thing. Other structures of argonaute proteins didn't show any eukaryotic insertion density, so we really had no idea what was going on. We did, however, know that changes in the argonaute structure occur upon binding to a target. And so in order to accommodate the target through argonaute kind of has to open up. So we get these conformational changes, but again, we don't know what's happening in the eukaryotic insertion. And so this is what I wanted to see, was could Hdxms be used to tell us information about the conformation of the eukaryotic insertion? Say, for example, could we see if there were helices that would be protected from exchange and therefore be visible? And would these change under these different conditions? Say when you had a guide and a target. So how does that affect the EI secondary structure or the accessibility? So are regions around the protein changing in different ways that might make it more accessible? And so this is what we wanted to test. And to test this, we turned to some collaborators at UC San Diego, Elizabeth Comici and Steve Saletti, who actually collected the data for us. And then they helped me do the interpretation, which was a lot of fun. And I got learn a lot of cool skills. And it was especially great during the pandemic lockdown to have a lot of computational data to work with. So we take our argonaute and we label it. We dilute it in that deuterated water. And over time, the regions of the protein are going to get labeled. Those hydrogens, normal proteins, are going to be switched for deuteriums. At different time points, we're going to quench it. We lower the pH and temperature. We then cut it up. So we use like pepsin to digest the protein, get those different pieces. Our observable exchange is going to come from those amide backbone hydrogens. Because those ones in the side chain, when we go and we quench the reaction, they're going to swap back to being normal hydrogens, normal proteins. And so we're not going to see those, but those ones on the backbone are going to remain. And they will have exchanged more in those regions of the protein that are more flexible, more solvent exposed. Because being tied up in secondary structure is going to protect them from exchange. And being in kind of more protected regions, central, say, are going to protect them from exchange. And we're able to more easily differentiate them by taking different time points. We can compare the exchange in different portions of the protein and see how those might change under different conditions, such as in the presence of a guide or in the presence of a guide and target in the case of argonaute. So you can imagine setting this experiment up in multiple different ways, depending on what you want to test. So let's go ahead and look at what some of this data actually looked like. So remember that we can see data in a couple of different formats. This is showing you an individual peptide spectra. So for this one peptide, in this case, it corresponds to the amino acid residues 532 to 540 in the protein. On our x-axis, we have time. So we were taking time points at 30 seconds, 60 seconds, 120 seconds, and 5 minutes. On the y-axis, we have deuterium uptake. In this case, it's in Daltons. What we're going to see is at 0 minutes, this is kind of our baseline. We don't see any deuterium uptake. At half a minute, we start to see our envelope moving over. It keeps moving, it keeps moving, and it keeps moving. And we can plot these onto a peptide uptake plot to give us the deuterium uptake over time. Because all these peptides have different sizes and different amounts of hydrogens, it's useful to convert this to a fractional uptake rather than just a raw uptake. And this is going to allow for a more easy comparison. So you can see that the maximum amount of uptake we get after 5 minutes is about 50 to 60%, or 0.5 to 0.6 fractional uptake. We can then plot these onto the structure, the crystal structure of the argonaute protein. Here we're using a colorblind safe color scheme, and so purple is going to be your most protected, so the least exchange, and yellow is going to be the least protected, the most exchange. Sometimes you might see a blue to red scale, with blue the most protected and red the least protected. But in this case, we're using this colorblind safe scale, and so here you can see you're kind of a middle amount of protection, which makes sense because you kind of have some of this hydrogen bonding, keeping the secondary structure of the alpha helix. If you look at this more flexible region, however, well, here you see you have a really high uptake. And so you have almost full uptake in the fragment after 5 minutes. And even at 30 seconds, you already have about 70% uptake. Whereas in regions that have a really strong secondary structure, maybe are a little more solvent protected, maybe a little more central in the protein, these are going to have a very low exchange. And so you can see that this fragment here is going to be very well protected. You can do this for all the peptides on the protein that you have coverage for and plot it over the protein. You can see that with the RNA-free protein, there are a lot of regions that have relatively high uptake. What about when you bind to a guide? Well, here we see that the relative uptake is going to be lower. We see more of the purples and blues and less in the yellows and greens. There's less dramatic changes when you bind to the target, but you still do see some changes. Let's look a little more specifically at what we can see in the peptide level. Here we're going to switch to a red-blue color scheme, where red is going to mean that the guide bound is less protected, and blue is going to mean that the guide bound is more protected than the RNA-free forms. Where might we expect to see differences? Well, you can see that there are differences over pretty much all of the protein, with the guide bound being more protected over most of the protein, and there's some regions where it's a lot more protected. One of these regions is the 5' binding pocket. So where argonaute holds onto the 5' end of that RNA guide, that microRNA or that small interfering RNA, that siRNA. You can see that it has a relatively high uptake with the RNA-free form. However, in the guide bound, it's almost completely protected from exchange. And this is because those hydrogens in the backbone, instead of being solvent accessible and freely exchanging, they're actually probably going to be involved directly in binding to that RNA. We see similar but not quite as dramatic effects in the 3' binding pocket, where you have protection being offered by the guide binding. And overall, we see that the guide binding is going to help certify the argonaute protein and introduce more kind of secondary structural elements, likely protecting the protein from exchange. We can map these changes, get the most granular detail if we have overlapping peptide fragments. This is one of the reasons why some people might want to use methods that, instead of using protease digestion, they use energy digestion methods in order to get a little more unbiased peptide fragmentation so that you can get more overlapping fragments, get most information. Some regions you might not even get any information depending on how the protein gets cut up. But the more pieces you have, the more information you can get. And you can see that here we're showing the difference, the delta du. It's a difference in the uptake between the guide bound and the RNA free. And when the guide is bound, you're going to have a lot more protection from the exchange over most of the protein, especially in the mid domain. What's really cool too about these experiments is that it provided the first more detailed structural information about the changes that occur in argonaute upon binding to a guide. So the Joshua Tor lab, they had earlier found that there were big changes in the protein upon binding to a guide. And they could tell this because if they use limited proteolysis, so they take a protease, so one of those pet protein cutters, and they add it to the protein when the protein is in the presence of the RNA or absence of RNA, and then they kind of take samples over time. The protease is going to have an easier time chewing up the protein if the protein is more floppy, but if the protein is kind of more sturdy, it's going to have a harder time cutting it up. And so you'll see more pieces more quickly if the protein is all floppy, and you'll see regions protected from the cleavage if the protein is more sturdy. And the parts where the protein is more flexible are going to be the parts that get cleaved first. And so what they found was that it was a lot easier to shred up the protein when it was RNA free. Then if you bound to the guide, it got very protected from the cleavage, only was getting cleaved at sort of the main points in between the domains. So this was some kind of key information that they found, and I followed up with my own experiments, testing different proteases, and I got very similar results. And then we added another layer of detail showing more about which regions of the protein in particular undergo dramatic changes upon binding to a guide. What about the effects of target binding? Here, the effects are going to be less dramatic, but we do see a couple of key regions, including regions that are known to be involved in changes in the binding to a target, which is a good validation of our experimental technique, such as the L1 region, the L2 region, that are known to have to kind of move to help the protein open up to accommodate for the target. We can map this out over the entire range of the protein, showing it under different conditions. And so you can see that data here in the supplemental of the paper. You also see that we use this 5XA, so that's the version with alanines in the place of those phosphorylatable residues. So that you can see here with the wild type, we weren't even able to get data for the eukaryotic insertion, probably because it was too heterogeneous. So it could be that the protein had some phosphorylation on it. Even though we tried to dephosphorylate it in vitro, it still could have some phosphorylation, things like this, that made it so we couldn't detect this. However, the 5XA was identical to the protein in all the other places, and so we use this 5XA as a stand-in for just the normal wild type. And that's just a technical detail. But I like to give some of these technical details because I know I have a lot of trainees watching, and that's the sort of experimental design thing that you might need to think about, the sorts of controls that you might need to have. What about the eukaryotic insertion? Because we don't have a structure of this, we had to actually model it in. And well, these all look pretty similar, right? In fact, what we found was that overall, the uptake in the eukaryotic insertion was not affected by the target binding. So you can see that comparing, so the region that actually covers the eukaryotic insertion itself is pretty similar in all three. The RNA-free, that's red, is a little bit higher uptake compared to the guide and the guide target, these blue and green respectively. But these blue and the green, the guide and the guide target, are pretty much overlapping one another. There's no difference between them. There's also no difference between them downstream. So at this point that kind of comes off of the eukaryotic insertion. However, in the RNA-free form, it is more exposed, it is more flexible, it's getting exchanged more. In the eukaryotic insertion itself, it has a pretty high fractional uptake, about half, and so this is suggesting that there's likely not very strong secondary structure there. It's likely very solvent exposed. There could be changes in the conformation that are happening in these different forms. However, because it's already at such a high exchange, we might not be able to really tell. Nothing too dramatic that we can detect with this technique. It doesn't mean that there aren't changes occurring. There just aren't changes that we can detect with this technique. But this is important information because it tells us that there might be other factors involved in making that eukaryotic insertion more favorable for phosphorylation. And so we suggest that there's actually potentially changes that are happening to make the RNA more accessible to interact with the kinase and things like this. And so more about that in the eLife paper if you're interested. Overall, we found that we couldn't detect any significant changes in the eukaryotic insertion upon binding to the target, but that region seemed to be pretty solvent accessible, pretty flexible likely, because of the high deuterium uptake. We did, however, detect conformational changes in argonaute that happen upon binding to the target, and we suggested these conformational changes might position the RNA and/or argonaute to interact with and potentially prime the kinase sequin alpha, leading to the hierarchical phosphorylation. So I did a lot of phosphorylation assays with a bunch of different mutants, trying to kind of figure out the order of phosphorylations. Ultimately, we came up with this hypothesis that then what's going to happen is when you have those phosphorylations, you have a bunch of negative charges that clash, leading to target release. Argonaute then phosphorylated is repressed from binding further targets. However, then the phosphatase PP6 can come in, dephosphorylate things, and argonaute can do it all again until CK1 alpha comes back. If you want to learn more, check out RE Life paper. But for now, let's just go back and review hydrogen-deuterium exchange mass spectrometry, or HDX-MS. So again, the point is that you're measuring the exchange of normal hydrogen, so that's your protium, your H, atoms for heavy hydrogen atoms, deuterium, or D, in a protein's backbone to learn about the secondary structure, so our helices, our sheets, etc. Solvent accessibility, so are these regions accessible to get wet? Binding locations of various ligands, or the parts where chains interact with one another, these sorts of things. You start with your protein in normal water, and then you label it by diluting it into diluted water. Your H's get swapped for D's, and the more flexible, the more solvent exposed the region is, the faster it'll get swapped. You quench the reaction by lowering the pH and temperature at different time points to stop the swapping, and then you use mass spectrometry in order to measure the weight of different pieces of the protein in order to figure out where it was heavier, where that swapping had occurred, and how quickly. In order to get that peptide level detail, you need to do some digestion, often with the protease pepsin, clean up the fragments using liquid chromatography, and then measure the mass of the fragments using mass spectrometry. This is going to allow you to do some different comparisons. Compare at the peptide level, so local comparisons, at the global level, so at the whole protein level. You can map to a structure if you know it. You can compare the presence of absence of ligands in different buffers, different other conditions. Lots of cool stuff that you can do with HDX-MS. It's important to know some of its limitations. So again, you're not getting the high level of structural detail that you would be getting from a technique like crystallography from cryo-EM. However, you are getting this information about dynamics, information about solvent accessibility, all these various things. So really cool stuff that you can do with HDX-MS, and there's so much I would want to do with HDMX if it was more readily accessible. But I'm really grateful I got to work with it in undergrad, and I hope that it becomes more accessible in the future and more people could use it. But for now, we can at least all admire the results from experiments with HDX-MS. And not just my own experiments, there's a ton of really cool studies out there that use HDX-MS to help figure out things that are happening in biochemical systems. And so I highly recommend that you check out these review articles that I will link to, and hope this example helped you see how useful it could be. Or at least just appreciate how cool chemistry really is, and maybe get a little more interested in argonaute. MicroRNA is really, really cool, but remember that argonaute is the one that's actually directing all that cool action. So go argonaute!